that is what is on these slides. I picked two random figures for my thesis to put as the figures for the front slide. So this is what signal transduction and receptors are all about. This is a protein. You've seen it before, probably. X-ray structure. And what it is, it looks like spaghetti. And in that spaghetti, you have helices, a secondary and a tertiary structure, protein. This protein, it's a KRAS protein, which I did my thesis on, and it was deemed to be undruggable. It's one of these highly implicated targets in cancer drug discovery. And this molecule, yellow and blue right here, peptide that was developed that actually inhibits KRAS, but it can't be given as a drug because the ADME properties of it are really bad. So the metabolism basically destroys it. It's really a tool compound for research purposes only. And I used it in my thesis. So basically, we have a binding event. Binding event changes the secondary structure of the protein, even tertiary structure. You can move different loops. This is each region of the protein is lighting. There's an alpha 3 here, like alpha 4, a P loop. There's switch 1, switch 2. There's different three-dimensional structures, domains. Well, these aren't domains; these are just subsections. But you can have domains, which are huge parts of a protein that have a specific function, like a kinase domain, things like that, that can actually be modulated by a drug binder. But once a drug binds, you're going to have an increase of certain groups of intermolecular force. It could be covalently bound. In that case, it's not coming off. You can have ionic interactions. You can have a lot of hydrogen bonds, which are a main factor in drug binding. You can have a lot of van der Waals forces, like hydrophobic interactions, which are usually common in deeper pockets of a drug. A great idea for targeting a receptor or a protein is you want something that on the three-dimensional surface has deep pockets where you can design a molecule to fit perfectly nice and snug and specific into that deep pocket. That's how you get a potent drug. But this protein, unfortunately, you look over the surface, there are shallow pockets, not very many divots that you can drug, which made it difficult for years, for over 30 years, to drug this protein. And the first drug that actually came out during my thesis that was FDA approved by a company called Amgen has a lot of side effects. Because if you make a drug that binds to one of these shallow pockets, it could, what's to say, it may not want to know the protein inside. May have off-target effects, and it does. That's the problem with these drugs. So anyway, that's what happens. You have this binding event, which changes the secondary structure, moves different things around. That causes the next screen, next secondary to change the interaction with this protein. Let's say like the next protein, which is RAS, it binds here. But once this binds, this alpha 4 moves over here. And it automatically fixes. Look at that. Like the alpha 4 moves like here. And RAF binds here. Well, now RAF can't bind there. And if it can, it's with a lower affinity than it would before. So that's an inhibitory effect. So these are the kind of things that are happening at a molecular level when you're dealing with drug binding and causing the downstream or interrupting a downstream pathway. So we'll learn about there's two types of drug interactions. There's a positive interaction and a negative interaction. One that stimulates a certain pathway, one that mitigates a certain pathway. So we'll learn about those different types too. And the way that the drugs are made for them are the same process. So we'll learn about that stuff. All right. So here's what we're talking about with the types of drugs that bind. So really we have different outcomes. So we have a drug, and then we have a receptor effector complex. You have a receptor that's on the cell surface. It has an extracellular domain, an intracellular domain. And then you have an effector protein. Let's say it's an adapter protein, like a receptor tyrosine kinase or something, right? Something that binds intracellularly that causes other proteins to then be recruited, activating them, maybe phosphorylating them, causing a downstream pathway. A drug can bind to that complex and disrupt it. It can bind to that complex, change the secondary structure of either the receptor or the effector, or it can bind in between them and block the interaction between them, or enhance it. You have inhibitor or activator. And then that changes everything downstream. So everything into the genetic expression. So all of these pathways modulate some type of gene expression, whether that be for an inflammatory response, or a metastasis, or apoptosis, really anything. They're all modulated by these pathways, or an action potential, right? In that case, ion channels. You can also have a drug and a receptor bind together, make a drug-receptor complex, which then can somehow modulate its interaction with the effector molecule. That can happen too, and that happens all the time. So it can either inhibit that or activate that. You can have a drug plus a receptor that make a drug-receptor complex, which then activate or cause a coupling of another molecule. So you can have and this is what some companies are doing right now, is you have a drug that can induce a protein to dimerize. So let's say you have, I'll give you an example. Well, I won't give you a specific example because I can't talk about it. But my company, let's say we have two proteins, or there's one protein. To be activated, it forms a dimer. So that means two of the same proteins in the cell, they're moving around, they come together. That activates them, changes the secondary structure a little bit, causing the downstream effect. What if, and this only happens when they're phosphorylated, each one of them is. What if you can mimic that phosphorylation and make a compound to bring both of them together? Then you'll be in business. You'll activate that pathway artificially. So that's what we mean by, we have a drug, binds to a receptor, it activates, another molecule comes in, another protein, causes that activation, and leads to a downstream effect. So this is all, making drug is an artificial human intervention into biological processes. And the last one, we have inhibition and metabolism. Yeah, inhibition and metabolism. If you inhibit metabolism, that's also activating the drug in an indirect way. But basically, if you don't want to remember all this, and we don't have to, there's two types of drugs. Disrupt a bad thing, like cancer, or activate a good thing, like p53, which is a tumor suppressor gene. Some mutations deactivate p53, meaning the cell cycle goes unchecked, the CDK cycling stuff. So if you develop a drug to inhibit that mutation, or to activate p53 in another way, or inhibit the proteins that break down p53, then, I know it's not double negative, but then that leads to a good thing. And you can cause a positive downstream effect or no cancer. So, two types of drugs, no bad thing, or we want a good thing. Not necessarily. It activates a thing. Hello. It's okay. Yeah, you so off-target effects or side effects means that instead of the drug binding to this specific receptor, it may bind to another one in the stomach or the colon or somewhere else. And that may cause something. It may not be good, it may not be bad, but it can cause a headache. It's benign. Most of these side effects are benign, but they're generated from these off-target binding events. Right. It's a lot of the chemotherapy agents are non-specific, but they get the job done in stopping the cancer cells from growing. That's one of the toughest things about making a cancer drug, is making it specific to a mutation type. So, right, because there's 1,000 mutations, but for a specific protein, there may be only 2 or 3. Or really, for most proteins, there's one that's prevalent and causes the cancer more than the other ones. Not 99, 30%. Yeah. I know that from my thesis. So that number gets thrown around a lot. There's 90% of pancreatic cancers are RAS modulated. But There was one paper that in the 80s that said 30% of cancers were involved in RAS, which is why it's one of these highly sought after drug targets. But then another paper said it was 12%. It really depends on which population you sample. So I don't know what percent. I say 30 to make my thesis seem more important, you know. But you're right, it's 90. Screw it, it's 90. But yeah, good. All right. So receptor theory. So receptors are the gateway into the cell. That is how the intracellular pathway gets started. Because we have the cell membrane. The cell membrane, yeah, it might be permeable to some drugs, but it's not easily permeable by every biomolecule. Like sugar, glucose can't get through the cell membrane quickly too well. That's why we need glucose transporters, GLUT4. So the receptors are the pathway. And the way those pathways are the gateway. The way those gateways are open is by ligands. You have extracellular ligands, whether they be hormones or growth factors or other things, other molecules or neurotransmitters, right? They bind to the extracellular receptor or extracellular domain of the receptor, which usually the receptor has an intracellular and an extracellular component. Once there's binding to the extracellular part, the secondary structure of the intracellular part changes to propagate that mechanism or propagate that change from the outside to the inside without the molecule, without the ligand having to diffuse through the cell. How smart is that? That's evolution for you. That's 4 billion years of evolution. Really smart. Once it gets down there, then you have certain secondary structural changes, which lead to the recruitment of other proteins, which then cause a huge downstream effect. That for every pathway, they're super complicated. We can go on for hours talking about one pathway. So we're not going to go. So in this class, we're going to cover the basics of the receptors and how they're activated, which is important to know. But we're not going to cover in depth about any pathway too much, because that's ridiculous. That's not good to ask. That's not on an exam, because in real life, I don't know. Like like for my research, I obviously do this, but like, let's say I'm on a project at work, right? I don't need to know every single drug partner or every single binding partner of every protein. I know the main ones from working with it for a while, for a couple of weeks. And then if there's anything else, I just look at a couple papers, a couple key paper review articles that have everything. So we need to know everything. We need to know the concepts, which is why I ask questions in the homework saying, list 3 ways or think about and explain 3 ways to do a certain thing. Because that's using your intelligence and your creativity and what you learn from the class and the different concepts. That's what gets you job, not spitting this down on paper. Although that might be impressive. Okay, that might get you a job. But once you're at the job, you won't do well once they switch over to another project. You need to use the concepts you learn to tackle anything. Okay. So yeah, this is what I mean by complicated. There's a lot of really this. Okay. Any figure you see showing a metabolic pathway does not show the whole story. This is all made by people, and each one of these figures you see online is made for a specific article. And they're made to show a specific thing. In this case, for GI-implicated cancers, we are showcasing different monoclonal antibody treatments, inhibitors of these key receptors that are involved in GI-implicated cancers. Also, downstream inhibitors that are affecting the intracellular portions of these receptor tyrosine kinase receptors, or even more downstream and more specific, which is RAF inhibitors. BRAF. Yeah, MEK inhibitors are thinking that's the case. There is a MEK inhibitor. But anyway, so and even going into the transcription factors, even going into inhibiting the cortisol, proteasome degrades proteins. So there's a ton of ways to intervene, and this is what they're trying to show. But is this really the story? No, because all of these factors and other pathways have their own proteins involved that are activated one after the other by certain mechanisms that actually interplay with this mechanism. So if you don't understand what I'm saying, that's good, because it's so complicated that it's tough to wrap your head around, at least the first time you're hearing. But once you see different figures, you'll see that they don't add up. Like, oh, what, this one had this one had this one from the grass, the dimer, what's going on, right? The other one has another pathway, the Wnt pathway that comes in and has some and activates these proteins also. What's going on? They all have interplay with each other. And they don't tell you that until you get to the workforce. You're like, whoa, I didn't know how difficult this was. So, like in my thesis, for example, there are, and it took me a while to learn it, but there are a ton of, so rats can turn on by another protein I'm showing you, which is the main focus of my work. There are several ways that there are several negative feedback mechanisms for the turning on of the protein that turns on rats. I heard that on my pipeline. I'm done. No, but I like teaching too much. I didn't get my degree to this. But it's really crazy. I'll just say that. I'll just end it on that. It's really crazy. But it's so interesting. I love this stuff because it's so interesting to learn. With every new paper that comes out, there's new discoveries of maybe elucidating some of these pathways. Yeah. Any questions? I know that was less information based, it's kind of a rant, but whatever. I was gonna say something. There's a lot of them, and a lot of receptors. There could be several types of receptors, and even isoforms of that specific type of receptor. The VEGF receptor, there's multiple types: VEGF1, VEGF2, that can both activate the same pathway. The difference between those receptors in terms of structure is minimal. They're 99% complementary to each other. So most likely, if you have a binder that binds to one, it'll bind to the other. So that's something to keep in mind. Because you might see that often, let's say wherever you go in pharmaceuticals, you may see, oh, I'm dealing with the 1, the 2, the 3, the 4. A lot of the times, most of them are very highly complementary. So, okay. All right, so I kind of mentioned this already, but here's how a transmembrane signal works, just to wrap it up in simple words. I'm going to read off the back because I can see it better. So you have a chemical signal crosses the membrane by binding to the extracellular end of the receptor, and then that causes an interest. Like, oh, well, there's different. Well, this is general. This is general. So let's look at the general here. A chemical signal crosses the plasma membrane and acts on the intracellular receptor. Okay. That can happen multiple ways. That can happen by a ligand, small molecule ligand, a growth factor, binding extracellularly, like we see here, causing an effect that changes the intracellular, and now the signal is inside. When we use the term the signal, that's not an item. It's not material. It's not a molecule. The signal. It's a change. It's a change that's propagated from the binding of a molecule throughout. It's a domino effect. So that can happen by that, or you can have hormones that are pretty nonpolar that can go through lipids, steroids, right? They're nonpolar. They can go through the cell membrane and directly bind to a receptor. That makes the job easier, less propagating, just binding. Then the drug binds to that extracellular. That's one way. Then you can drug. So then you can have a drug bind to the extracellular domain, which we talked about, thereby activating an enzymatic activity of the cytoplasmic domain, like we talked about. That's traditional receptor on the membrane bound receptor. Or you have the drug binds to an extracellular domain of a transmembrane receptor bound to a separate protein, tyrosine kinase, which activates. So what that means is You can have a receptor that's on the membrane that binds a drug, which then recruits another receptor or recruits another membrane-bound protein to activate, and that causes a downstream effect. And you don't have to memorize any of this. Just concept. Understand what's happening. You also have the drug binds to and directly regulates the opening of an ion channel. That one's completely different than the other one. The other one's kind of interplay with each other. You can have an ion channel, which is a specific protein that it has a channel through the middle that ions will come through: sodium, potassium, calcium, right? Or the main three. Chlorine. You have a ligand that binds to the active site of that protein, thus opening the receptor and opening the channel. And then the gradient, the ion gradient, is what controls the flow of ions through that channel. That's how action potentials are generated, which is your nerve impulses. We will have a drug binding to a cell surface receptor linked to an effector enzyme by a G protein. We'll learn about that later on. That's the last thing we'll learn, is G protein coupled receptors, which is its own thing. Okay, so this is just general 5 ways of signaling. Okay, so we're going to talk about 5 types of receptors. I'll just write them on the board. I'm not going to write down exactly what all of them are. I'm just going to. You wrote. Okay. So. Steroid hormone. Okay. So the first type of receptor is a steroid hormone receptor. Now, steroids are lipid-based. They're lipophilic. They can cross the cell membrane relatively easily. They can find their steroid receptor is free in the cytoplasm. It's not bound to the cell membrane like a G protein coupled receptor, like a receptor tyrosine kinase, like the sticks we saw before, or an ion channel. Those are all bound to the membrane. They're membrane anchored. You can have receptors that aren't membrane anchored, like cytokine receptors, and also this one, steroid hormone receptor. So what happens is steroid hormone will diffuse freely into the nucleus, into the cytoplasm, bind to the receptor. Now that the receptor is activating, it can then translocate into the nucleus. And the receptor, in this case, the steroid receptor, has a DNA binding domain. And it can act as a transcription factor and initiate transcription and then translation of key proteins that are involved. And let's say if it's an anabolic steroid involved in protein synthesis and growth. So that's how all these different receptors work, all these different steroids work. So the receptor itself is a transcription factor. So that's good to know. Simple enough, I think. Yeah. Any questions on that? Okay. So then we have here's another example of that, actually. So glucocorticoids. So glucocorticoid, steroid. So it's like hydrocortisone or prednisone. I mentioned this, I think, last class. So it binds to, so we actually have a transporter that brings it into the cell. It can't diffuse through the cell freely, through the membrane. It has this binding partner, the chaperone, called transcortin. But that goes away once it's in the cell. Anyway, once it's in the cell, it binds to the glucocorticoid receptor. And then you have this heat shock protein, which binds to the receptor in the absence of hormone. But once the hormone's bound, the heat shock protein is removed. What the heat shock protein does, and we've seen these before maybe, but if not, no problem. Just you learn something. Whenever we see HSP, that's heat shock protein. They are protecting proteins, support proteins, chaperone proteins. So they're proteins that protect other receptors. In this case, the heat shock protein binds to the glucocorticoid receptor in such a way that it protects its active site. It's low affinity. It protects its active site so that no degradation of the receptor could happen, so that the receptor doesn't misfold and block off its own active site. So that's pretty cool. And the receptor then is pretty proud of it. And these heat shock proteins they stabilize it, but they have a low binding affinity to the heat shock protein, or to the glucocorticoid receptor. Now, here's a question. Why do you think this support protein would have a low affinity for the receptor it's trying to support and protect? Why do you think it would be a low affinity? Wouldn't you think it would be a high affinity? Why a low affinity? Anyway. Okay, think about it this way. What if it was a high affinity? What if it was nanomolar binding? Would that be able to happen? What do you mean? Elaborate. Okay, good. So everybody else, that's a good start. Do you have a thought? Not necessarily, but that's good from looking at it from an energetic point of view. Any other ideas? Before I kind of like push and tell you? Okay. Well, anyway, if it was a really high potency towards this receptor, it wouldn't be able to come off. If it doesn't come off, the steroid can't find the active site. It's being blocked by the chaperone protein. It needs to be a weak enough interface between the heat shock protein and whatever protein is protecting, whatever receptor is protecting, in order for the natural ligand or a drug to kick it off and bind to that active site. So if you have the steroid, which is a 1 nanomolar binder, actually, since we're dealing with it, if it's a natural steroid, it's much lower than that. It's picomolar binder. If you have a picomolar binder, and then you only have, if it's like the kb is 10 picomolar, you end up with 20 picomolar. And then the heat shock protein, you have 100 nanomolar of it, more, but its affinity is only 2 micromolar. It's like, all right, I'm binding to this, I'm protecting this protein, but if anybody else who really wants it, they can have it. Then steroid comes in, go away, binds to the receptor at a really tight binder, and that causes a downstream effect. So yeah, that's the way chaperone proteins work. Yeah. And then what this does downstream is we always say if whoever's drawing figure is too lazy to draw the rest, they just say alter transcription of specific genes. And there's a lot of genes. That's gene therapy. You want to focus on all the different genes that a specific pathway involves, and there's usually a ton for changes in expression of. So you have lower COX-2 and higher lipoprotein. So COX-2 is what is inhibited by aspirin or Advil, or acetaminophen also. They are COX-1, COX-2 receptor inhibitors. Or so this is an indirect way of lowering or of inhibiting COX-2, not by on a protein level binding to it and inhibiting it, but by actually stopping its gene expression. So this is what we mean. You have 2 sets of anti-inflammatories with 2 completely different mechanisms. So think about that with questions like the atenolol question of ways to stop its toxicity. You can think about it from a genetic point of view. You can think about it from a protein point of view. You can think about it from a metabolism point of view. You can think about it from a clinical point of view. And you can have answers for all of them. So as drug discoverers, if you want to get into that, That is the mind you have to have. And that takes years of dedication and exposure to this stuff. When I go to work, I feel like I'm the dumbest person there. Because I am. But I'm happy I'm the dumbest person there. Because I learn so much every day from these people who have like been doing this for 20, 30 years. And they look at it from all these perspectives at the same time. And I'm still thinking like, okay, wait, you got the receptor, and you got, oh, we're looking for this, we're looking for that. They're like, oh, what about the metabolism of Psych3AYP? I'm like, I'm not even there yet. I didn't even think about that yet. But like that's the kind of place I want to get to. They think about all this at once. So crazy. But anyway, that's how that works. Intracellular receptors. In terms of action time, so hormones produce their effect, but they have a lag. These hormones. The reason why is because hormones, according to these steroid hormone or these hormone receptors, the hormone receptors are transcription factors. That means they have to initiate transcription and translation, protein synthesis. That takes time. So you're going to have an onset or an offset of 30 minutes to hours, depending on which hormone it is that you're taking or which drug you're taking that affects that hormone receptor. So as opposed to other drugs, which can like anything that has to do with nerves, you can turn off an action potential like that in milliseconds. Or if you take the drug, it might take a minute, right? So this is very simple. Yeah. Okay. Here we have the next type. Any questions? Any questions? No? Okay. Next type is RTKs. My favorite. They're called Receptor, tyrosine. Now, does anyone want to take a guess at what it does based on the name? So what's tyrosine? Right, it's an amino acid. What's kinase? You know what that means? No, enzyme cuts stuff, but what's some kind of lyse? That's a lyse, something like no, lysate is like a cell lysate. Lysate. If you're lysing anything, you're cutting something. But kinase. Good. Yes. So kinase is adding a phosphate. So is it another term? Another term you should know. So kinase adding. So if you have something that's a kinase, it adds a phosphate to the substrate. Then you have the opposite of kinase. What's the opposite of kinase? Or something. Close. Closer. Don't become a phosphatase. It could be a dephosphorylate. That's cool. Yeah. Dephosphorylate. So usually, if you protein or you take the downstream proteins that are activated, most of them are activated by phosphorylation, and they're deactivated by dephosphorylation. Happens by a phosphatase. So that's just two antagonistic terms. Other negative feedback mechanisms for receptor tyrosine kinases have to do with phosphatases taking off the phosphates. Also, what's the tyrosine part? The kinase is phosphorylating a tyrosine, a receptor tyrosine kinase. So you're phosphorylating a tyrosine on a receptor. That's exactly what you're doing. And that's what the receptor is. That's why you have this big block. Right? Why is the 100 code for tyrosine? So the way this, I'm not going to read all this stuff, but the way this works and you have the receptor occurs in a dimer. You're never going to have one of them around. They might be, if you're three-dimensional, let's say you take the cell membrane, flip it over, one of them might be here, one of them might be here. The cell membrane is like when you were little, like a ball pit, like a amusement park or something, right? It's like that, where if you look at a ball pit, you're like, oh, it's really crowded. Well, you could walk pretty easily. That's what the cell membrane is like for these proteins. If there is a chemical force or some kind of recruitment mechanism that is attracting one of these dimer partners to the other, that is going to bring them through the ball pit and connect. Even though there's stuff in the way, the phospholipids will move. They're easy to move over. So, how does this happen? Well, you add a ligand. You add an extracellular ligand, or ligand, how do you want to say ligand? Sounds cooler, and you say faster. So, oh yeah, there's a ligand. Oh, the ligand. I said that perfectly did all. But you said how cool. But anyway, so the extracellular ligand is EGF. Simple one. It means extracellular growth factor. Very standard growth factor. So you have it binds. It can either bond. This is one representation of it, where you have two molecules. One of them is binding to each monomer of the receptor, or you have this other one, which is all the same type. Where there is one, there's two dimers. Then you have one ligand come together. So like each monomer is both are relevant in their own way, depending on which receptor tyrosine kinase we're referring to. So anyway, once they do come together, they phosphorylate. They're kinases, right? But where is the other protein? Like, what's it doing? Does anyone know what it's doing? Right, but it's not dephosphorylating. Phosphorylating. That's where the other phosphate goes. That's the first. So phosphorylates itself. The receptor, so each monomer of the receptor phosphorylates the other one. So call this autophosphorylation. Meaning technically the receptor is one thing, and it each domain of it phosphorylates the other domain. Wait, do we have to wear masks though? I should be loud. Do we have to? Follow. But in a few weeks, I think it's cool. Right, but I mean, the colleges will soon follow. Probably. Anyway, sorry. Feel free to put up. But the autophosphorylate. The phosphorylate comes on tyrosine. The tyrosines and serines and threonines. That's it. I'll write those. Tyrosine, serine. Those are the three amino acids that are frequently phosphorylated. So you have two types of kinases. There's a tyrosine kinase and there's a serine threonine kinase. The reason why there's not three kinases is because the serine threonine kinases they can phosphorylate either one, but there's different types of kinases that are serine threonine kinases that are involved in several pathways. Yeah. So there's 517 kinases that are characterized in the genome. They fall into these three categories. And then there's more categories after that. But there's a general three categories. Or two categories technically. Yeah. Okay. So anyway, after the phosphorylation, What can happen then is we have substrate turns to substrate product. Things B. Now phosphorylate the substrate. That substrate, there's a ton of them. So another way to represent this. On the answers that I have are other or B.
Hi, Professor. We can't hear anything. Yeah, usually when you turn your back and you're writing on the board, it's very hard to.
Oh no. Okay. Oh, there's someone in the lobby. Oh, I didn't even see that. Oh, okay, they're in. Okay. To the board now. All right, cool. Can you hear me now? Can you hear me?
Yep.
I guess I just went away from the board, so it was away from the laptop, so it was tougher.
Yeah, we can hear better now.
Okay. All right, good. So these adapter proteins can come on and stick to the phosphorylated sites on the receptor tyrosine kinase, leading to whatever downstream effect. So all you guys need to know for receptor tyrosine kinase, the second type of receptor, you have a ligand, it binds to the receptor, causes dimerization, followed by autophosphorylation, and then some other downstream effect, whether that be adapter proteins bind or whatever. You don't need to know specifics after that. Just know how it's activated. And there's different ways to prevent this activation if we're talking about drugs. We need to always think, whenever I go over one of these purely biochemical things, we need to think about how it can be drugged, right? So we can make, or quick, let's go over some ideas. Who has an idea of how we can drug this and disrupt it? So how can we drug it and disrupt this pathway?
Professor, you could possibly inhibit the binding of the EGF in the first place?
Yep, exactly. So you make it competitive. Yeah, you can have a competitive EGF receptor inhibitor, like a monoclonal antibody they have for that, and they also small molecules. Great, that was Ben, right? Good job. What else? Right. So in different types of kinases, not these ones. These are just tyrosine kinases. Different types of kinases. You have serine and threonine to also be phosphorylated. Mm-hmm. Yes. So what's one other way to block this? Ben said the most common one, which was blocking the receptor from binding to the ligand. What's another one? What if you can't? In some cases, you can't because whenever you are competing, this is just a rule of thumb that's really tough to battle. That if you have a natural ligand and you're trying to compete with it in its own active site, that's very difficult. You have to be extremely potent because the natural potency of this EGF to the receptor is nanomolar. So you have to make a drug that's nanomolar or better to be able to compete with it. But what if you can't compete with it? What else can you do?
Is there a way to prevent the monomer from traveling through the cell membrane?
I'm sorry, can you repeat that?
Sorry. Is there a way to prevent the monomer from traveling through the cell membrane so it's almost locked in place?
Good idea. You're closer, but there isn't a way to do that. Good idea though. I like that. That is actually wrong answers like that are really good because you're thinking about the system, which is great. But on that same note, if they don't move, they won't find each other, right? But they can move. Well, what else can you do? That's perfectly feasible. Yes. So what if you knew the secondary structure of this is a protein, remember? What if you knew the secondary structure here? It's off a helical, let's say, right? What if you're like, there's a drug that binds here and it prevents the binding to the other dimer partner? Sure, completely viable. I don't know if there's any drugs like that out there. There might be. I don't know every drug. But yeah, definitely. Good. I like these thoughts. Okay, moving on. What? Oh, there we go. Okay, cool. Oh, here's just another example of receptor tyrosine kinase with insulin. So insulin, on the bottom of the screen. So insulin binds, or insulin receptor binds insulin. An insulin receptor is a receptor tyrosine kinase. So the same thing happens, even though they're all doing different things. So insulin is completely different than EGF, an extracellular growth factor, causing RAS activation, right? Completely different, but it's the same type of receptor. It activates the same exact way. So if you have a scientist that they're an expert on receptor tyrosine kinases, they can they can be leads on a bunch of different drug projects. So you can either be an expert on a receptor or a pathway, or you can be an expert in a disease. Most clinicians are experts in diseases. Biochemists and drug discovery and pharmacologists might be experts on GPCRs or kinase inhibitors in general. My advisor was the head of kinase research at Merck for like 20 years. So he was a kinase guy. So he knew everything about kinases. And he's who I learned kinases from. So hopefully I'm making him proud. No, I'm probably not, because I'm not going into super detail. He likes detail. Insulin, they get a bunch of different pathways. And what they do is they activate the force that will lead to the opening of group 4. Group 4 is a membrane channel protein that allows glucose to come into the cell from the blood. This is how blood glucose is decreased by insulin. There's more to it, but this is the simple mechanism of how it happens. And we'll talk about that mechanism when we cover diabetes more in depth. So, yeah. Any questions on that? Then we have cytokine receptors. Okay. Now, these ones are similar to receptor tyrosine kinases, but they don't autophosphorylate. So read this in a second. These receptors use a mechanism closely resembling their receptors, except that in this case, the protein tyrosine kinase activity is not intrinsic to the receptor molecule. What this means is that you have two dimer partners, but when they bind together, there's no autophosphorylation. They just sit there. No, they don't just sit there. They activate. What that activation does, again, it always stems from a change in secondary structure. A loop moves over, and helix moves over, causes a conformational change that is specific for the binding of another protein. In this case, this protein is called JAK, K-A-K. So, cytokines are usually promote inflammatory responses, inflammatory cytokines, right, involved in the immune system. So you have the cytokine binds to the cytokine receptor. The cytokine receptor is activated, and then the activation of it recruits this protein called JAK. Another big pathway in immunology, which I'm not an expert on by any means, is the JAK-STAT pathway, and it's involved in cytokine response and the different T cells. Yes, it's involved in this. Not off the top of my head, but it's this, but a lot. You want at the end of the I did watch a video on that. Let's look it up at the end. That's a good one. Okay, so basically, to activate what the cytokine does is it activates the receptor, which then dimerizes, causing JAK to bond. JAK then, what do we think the K in JAK stands for? Kinase, right? Janus activates. Am I right with that? Yeah, Janus kinase. I think it's Janus activating kinase. Anyway, so this kinase then phosphorylates the receptor. Now it looks exactly the same as the RTK. Once it's the same as the RTK, similar things can happen, meaning proteins will bind to those altered phosphates. Proteins such as in this case, now, STAT is not related to statins, so it's completely different. STAT is an intracellular protein that brings upon immune responses. It can be, once it's activated, it can dimerize, and it can become a transcription factor, which leads to the gene expression of cytokines, more cytokines, and just different immune responses. And there's a ton that can accelerate different responses. Like a lot of and receptor through the JAK-STAT pathway. And there's different stats. There is STAT1, another one. That's a similar thing. So, is that the cytokine is different from the receptor tyrosine kinase because it doesn't autophosphorylate. It requires JAK to phosphorylate the tyrosines on the receptor, which then leads to STAT binding and then downstream activation. And then once the activated STAT comes into the nucleus, it can then regulate gene transcription. Yes. So also interleukins and interferons are also modulated by this pathway. Okay. Any questions? All right. That's basically what we went over. Ion channels. The fourth one. Ion channels. I'll put a bar next. They are the fastest one, meaning they have the fastest activation time. Something bind a ligand binds, the channel opens, the protein or the proton maybe, but the ion comes in or comes out based on the ionic gradient. The change in this ionic gradient determines whether a nerve cell is polarized or hyperpolarized or depolarized. So it can change nerve impulses like that in milliseconds. So that's why ion channels are different. But here's basically how they work is you have a picture. Here's a good picture. Kind of like a poem. Really a lot, but mostly parasympathetic nervous system. So everything that's involuntary in your body is regulated by acetylcholine. The other ones, these are on the order hormone receptors in the order of minutes and hours. Receptor tyrosine kinase, cytokine receptor. Well, cytokine receptor. Well, yeah. Well, steroid hormone is really hours. These two on the order of minutes, and then ion channels are on the order of milliseconds. And also they have different subunits too. And each subunit has a function. So you see there's alpha. It's not just one protein that's, well, it is one protein, but it's not one protein that's shaped like a cone, with like a hollow cone. It's multiple domains, not to be a cone. And these domains have specific functions. Like one of them could be the acetylcholine binding domain. One of them could be the lipid tethering domain, right? Or maybe downstream there might be another binder that turns it off, like a phosphatase domain or something, right? So you have these different domains. And yeah, and that basically modulates the opening and closing. So you can have inhibitors for these. So if you like, acetylcholine is essential for all involuntary neural synapses and also memory, really everything. If you have low acetylcholine, it leads to Alzheimer's, or it's one of the biomarkers for Alzheimer's. Curare is a drug that inhibits acetylcholine binding, which can cause paralysis. Why would you want to do that? What if you have too much acetylcholine? Then you might want to, too much acetylcholine leads to Parkinson's. Too many like involuntary movements and like tremors and stuff like that. So you might want to use a drug that is a weak inhibitor for acetylcholine, so then it can kind of mitigate those that are overexpressed, that over activation. Okay, but overall inhibition can cause death if you inhibit the ion channel. Yeah, so here we go. The time elapsed between binding of the agonist to the ligand-gated ion channel. These are called ligand-gated ion channels. There's also voltage-gated ion channels, which are also present in nerves. They're measured in milliseconds. Yeah. Okay. Any questions on the ion channel? No, that's the fourth one. We're ready for the fifth one and final.
Professor,
the GPCR.
Hello?
Oh, yes.
Yeah.
Is there a question?
Yeah, could you go over that again just for 1 minute, please?
Sure. So, in essence, you have a extracellular ligand like acetylcholine or maybe another neurotransmitter that binds to the closed ion channel. Once it binds, it opens the ion channel, leading to an ion either coming into the cell or leaving the cell, dependent on the ionic gradient inside and outside the cell. This is more common in neurons than any other cell type. Does that answer? Okay.
Yeah, that's fine.
Okay. Thank you. Thank you.
No problem.
Okay. The last one and the most complicated study, one of my favorites. This is my second favorite. Receptor tyrosine kinase is my favorite because I studied it and it was the easiest. My second favorite is the GPCR. G protein coupled receptor. It does a lot. They can do a lot. It's responsible for adrenaline. It's responsible for alcohol. It's responsible for a lot of things. Opioids. And more. Many, many things. I'm too tired to think of more things. Okay. Basically, what is coupled protein receptor? It is a receptor, a bunch of squigglies on the cell membrane. It has 7 transmembrane domains. What that means, transmembrane, across membrane. They are membrane is both now have an extracellular receptor base Both are that are membrane bound. Need to functional capability to bring the signal from the outside of the cell to the inside of the cell. So that's how the GPCR is no exception to that rule. Now, this is not a second part, right? What's the G-coupled protein? Aha. Well, the G-coupled protein, three proteins. The G-coupled protein that binds to the inside of this coupled proteins. So it's crazy that I just thought of this now. GPCR has 4 letters. There are 4 proteins involved in a GPCR. Wow, that's a new way I'm going to teach it. I like that. We'll go over what it does in a second. But yeah, so here's just my point. For those of you online, you can just look on your screen. The inside of the receptor, we have it's called G alpha, beta, and gamma. Those are individual protein subunits that bond together in a complex and bind to the intracellular part of the receptor. That's the 4, GPCR. You have G alpha, beta, gamma, and the receptor. That's all 4. Now, questions on that? Okay, the next step is what they do. We'll go back a slide. Oh, well, that's a great slide. My advisor made that slide. But we'll go back. Okay, we'll go back to what they do. Well, here's what they do. So you have the ligand and binds, causes the activation of the GPCR. This then causes GTP to bind. I don't like the way this. You have GTP binding triphosphate, very similar to ATP, adenosine triphosphate. So GTP is another energy molecule. It donates a phosphate when it works in a reaction. Like a GTP to a GTP. But anyway, the activated G protein will bind GTP to turn into GGTP. G protein bound to GTP, which will then cause another downstream effect of activating an effector molecule. This activating the effector molecule is phosphorylates that effector, or it just uses the energy to bind to that. Then once the effector is activated, the phosphate comes off, and then the cycle continues. So let's put it this way. That didn't make much sense, but I'll say it differently. The G-coupled protein receptor is in the off state when it's bound with guanine diphosphate, GDP. That's why the G is starts as G, GDP. And you have that little protein complex of the alpha, beta, and the gamma bound to GDP, guanine diphosphate, like ADP versus ATP. ADP is lower, it's not really used for energy. It's the byproduct of the energy from ATP. You have ADP plus P. Very similar concept. You have GDP, right? Once the receptor is activated, GDP comes off. GTP from the inside of the cell now can bind. Once it's bound, this G, and it binds to the G alpha. So that's important to remember. So GTP binds to G alpha. Once the G alpha is activated, it can then propagate that effect throughout other signaling partners, which we'll cover. We'll cover one big one or 2 big ones in a second. Then once that happens, the GDP gets phosphorylated, or gets cleaved to become GDP plus P, and the P comes off because then that goes into the biosynthesis, makes more stuff. So that's generally how it's turned on and off by GDP, which would modulate it going from off to on. So now let's take a look at exactly what happens after that in more detail. I really like this chart. We're almost done too. We have 10 more slides. 7:30. We'll be out of here by 8. So we have this is the GPCR. A little bit different orientation, different way of writing, but whatever. You have these are the beta adrenergic receptors. You have an agonist to this, an epinephrine agonist. It leads to an increase of heart rate. You have why am I blanking? Blood vessel dilation. Sorry. You have blood vessel dilation. You have pupil dilation. No, your blood vessel constriction. Sorry, your blood vessel constriction would increase the blood pressure. Right, you get more oxygen to the tissues faster than when you constrict the blood vessels. So basically, this is fight or flight. When this is turned on, you have a sympathetic nervous system response, a fight or flight. Once it's activated, it then catalyzes the exchange of the GDP coming off, the GTP binding the G alpha S subunit. Now we're throwing an S in there, but it's the same concept as an isoform. You have G alpha S, G or GS alpha, GS beta, GS gamma. I'm used to saying G alpha S, but whatever. And this GPCR is lipid tethered, meaning once this GPCR, the receptor, is activated, these G proteins are not just coming from the cytoplasm and just being always, my body's activated, I'm going to go hang out with them. No, they're in the membrane moving around like the ball pit, right? Just from the inside. And once the receptor is activated, there's a chemical reaction that causes them to attract each other. There's a conformation of the receptor that is complementary to the binding site on the G alpha S, which causes that interaction to stick. So that's one way to block this interaction, right? If you were thinking of a drug, you can block this interaction by making a drug to prevent receptor binding from the G alpha. Once it's activated, that's alpha in the form comes off of the complex. Is the next part. Comes off of the complex. Now here we go. Activated GSA separates from GS beta gamma, moves to another membrane protein called adenylyl cyclase. Tough to say. A lot of Y's in there. Adenylyl cyclase. Cyclase, what do you think that means? It cuts something that has to do with cyclic, right? That's what it does. So we'll take some of that. Adenylyl cyclase. So adenosine cyclase. That causes the formation of AMP. Cyclic AMP, cyclic adenosine monophosphate. So after this GS alpha is activated and it has a GTP bound to it, it can move over to bind to another membrane protein called adenylyl cyclase. Activating adenylyl cyclase to convert ATP to this super important second messenger called cyclic AMP. Now, we didn't really cover first messenger and second messenger. I think that's a question on the homework. But a first messenger, I see everyone look up. It's all homework. First messenger is before the signal amplifies. We talked about signal amplification. Before the signal amplifies, the one molecule that gets this all started is called the first messenger. Whether it be a hormone, whether it be an intracellular growth factor, epinephrine, whatever the drug, whatever it is, it's the first messenger. The second messenger is after the amplification effect. So this epinephrine can only activate one of these. The adenylyl cyclase that is activated by one binding can transform ATPs over and over again into cyclic AMPs, and we have now 10,000 cyclic AMPs from one epinephrine. That's what we mean by amplification of signal. Now those 1,000 cyclic AMPs have different responses. You can either have the activation of protein kinase A, that leads to the cellular responses caused by epinephrine, meaning the increased blood pressure, the increased heart rate, the sweating, and all that stuff. Increased skeletal muscle contraction. Or it leads to other effects. It can also be degraded. It also leads to a plethora of other effects depending on the cell type. So, in terms of what we need to know, this is it for the GPCR. Understand this pathway, and then know after signaling AMP, the second messenger is made, a lot of things can happen. Know that maybe, for one example, the epinephrine is an example. So it can lead to activation of the sympathetic nervous system. That's a good thing to know. But also a lot of other stuff. So here's the outcomes. Differentiation of T cells, and you have immune responses, interleukins, chemotaxis, histamine response, mucus production, proliferation, a ton of stuff in different types of cells. You don't need to know all this stuff. You don't need to know really any of it, but just know that cyclic AMP is a really, really important second messenger. So if you're thinking, how can you modulate a GPCR? Why would you want to modulate a GPCR? A ton of reasons. You can affect all these different things. But I wouldn't want to affect a GPCR because that can make too many downstream effects. If I want to be more specific, maybe I want to target because there may be other effects that are cellular that are good, that are the outcome of the activation up here. But protein kinase A might lead to some kind of neurodegenerative disease. Who knows? If it's mutated. So I want to develop an inhibitor for there. But it won't inhibit the production of cyclic AMP, which is important for, like we saw, a ton of other processes. If I make a drug for protein kinase A, it will only inhibit the protein kinase A responsible pathways after cyclic AMP is produced, which may be valuable for whatever disease. That's something else I want to mention, that developing a drug that has high specificity and low toxicity usually occurs if you make a drug that is more downstream of a pathway. Because as we saw in all these different pathways, we'll go all the way back. If we make a drug for EGF, that much stuff never does. We'll die. If we make a drug that's for a non-mutated EGF, a drug that belongs to a wild-type EGF will die because we ourselves can't grow. But if we make a drug that's really specific to her, that will only impact the transcription involved with her, which in cancer cells have to do with cancer. So that's something that one of my PhD colleagues was developing an ERK drug. She did, and with other people also, and it was a lower potency, but it was still a really good proof of concept experiments there. All right. So, any questions on the mechanism of the GPCR? Because this is probably the most important part for this receptor. No? So one more thing about GPCRs is that they have a really complicated interplay that leads to its own negative feedback loop. So I did mention there are different types of G alpha subunits. Maybe I did. But anyway, there are 3 different types. There is G A. Is G alpha? Well, I guess there's 3, but we're not going to focus on 2. Well, there's different receptors, but let's just focus on. All right, we'll be okay. We'll do G alpha S versus G alpha I. There are 2 types of so remember, G alpha. It's the one that binds to the GTP, and it moves across the membrane, binds to adenylyl cyclase, causing the cleavage of ATP to cyclic AMP. But that's G alpha S. G alpha S subtype. TPCR, this is a beta 2 adrenergic receptor that's more focused on lungs, on increasing respiratory capacity in the heat of battle or when you're running away from the tiger, right? So, but TPCR beta 2. Oh, by the way, an easy way to know the beta 1 versus beta 2. Beta 1, you have one heart. Beta 2, you have two lungs. So beta 1, responsible for increasing heart rate and increasing force of contraction and constriction of blood vessels, etc. All heart-related things, circulation. Beta 2, when it's activated, increased respiratory capacity and respiratory rate. Okay, so then what did G alpha X activated? Adenylyl cyclase, CAMP, protein kinase, and activates other signals. We saw that. What if we want to shut this mechanism down? We can wait until the cell is exhausted of GTP, right? Or we have the activation of another GPCR that is post-translationally phosphorylated. This might happen in a response to too much GPCR activation. The expression will also lead to proteins like other GPCRs that are phosphorylated. These phosphorylated ones, think about them as like the evil twin. They will actually work and activate a different G protein called G protein I. This G protein I will do the exact same thing. It will bind GTP, but then it will inhibit adenylyl cyclase. Instead of activating it, it will inhibit it, leading to the deactivation of the entire pathway. Does that make sense? All right, good. Now, what doesn't make sense is think about it. Think about this. For those of you who took CHEM-2, right, you're in CHEM-2. This is an equilibrium. There's always in every one of these on versus off things where you have a negative feedback loop versus a positive activation. They're all happening at the same time, but there's an equilibrium between them. At time 0, where nothing is happening, there is a slow forward reaction of the cyclic AMP being activated for transduction. And then there's a slow amount of GPI that is inhibiting that adenylyl cyclase, going back and forth, back and forth, back and forth like this. Without a stimulus, without a ligand binding, that happens. It's at like 3%, both ways at 3%. When there's a ligand binding, beyond the G alpha S subunit activation, adenylyl cyclase, blah, 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 goes like that, 100%. And then following that, because we know the genes that are expressed from that activation, some of them are the evil twin. And they will have a time delay, and they will start to be expressed. And in this pathway, will start to speed up. And eventually, everything will come back down to that baseline. And the ligand will probably be released. So there's a lot of interplay between on and off rates of any mechanism. Otherwise, we wouldn't have regulation. Regulation is a really important word for signal transduction in any mechanism in our body, any metabolism in cells, regulation. If we don't have regulation, we get diseases, we get cancer, we get autoimmune diseases, we get a lot that can happen to us. So, yeah. Any questions on that? No? Okay. I'm talking about CAMP. It's important. Just know it's important. For cellular outcomes, it's important. So, calcium is another second messenger that's important, but we're not going to cover it too much. If you want to learn it, so I have if you see the slides, I have an article that is in the notes section of it, where there's another pathway that's activated by a GPCR. We have these to the influx of calcium into the cytoplasm from the endoplasmic reticulum, the ER. And this causes a ton of responses also. You have calcium is involved in muscle contraction, calcium is involved in bone marrow and stuff like that. So also calcium can also bind to protein kinase C and lead to the pathways. So calcium is another one of these second messengers that is super important for drug discovery. Knowing calcium is important. And I have actually a paper coming out on, I don't know when it's coming out, but it's in submission, of talking about how calcium and RAS, like we talked about before, have some kind of interplay that was previously not really discovered. So once that comes out, I'll send it. It may take a long time. I don't know when they say it's older, but we'll get your paper back in 2 weeks, and like with review with our nope. Hopefully semester won't be over. But anyway, that's for your own reading afterwards. The only thing, so here's a discussion question. Yeah, ignore the Blackboard part. That was for, I took this question from my other lecture from Keene University. They use Blackboard. So what I want to do is I want to talk about really quick, and we can watch a video, and that's it. On, oh, we have a lot of time. Okay. So what we can do is talk about RNA interference. Really cool mechanism. Who's ever heard of RNA interference? Oh, perfect. Oh, you've heard of it there? Yeah, exactly. So not many people though. So that's good. It's really good. So it's a relatively newly studied mechanism of how using RNA we can silence genes and use it as a drug to silence genes. Now, we may know RNA-based drugs like the vaccine, right? That's RNA-based. That does something different. That just translates for a mimetic of the spike protein, which creates our antibody. But RNA and also DNA is a powerful tool in drug discovery. So what happens is, I'm not going to go through the entire thing. I'm not going to test you on this. It's just really good information. Double-stranded RNA that is coded with the complementary strand of a specific mRNA sequence that you want to remove. The way that works is you introduce that double-stranded RNA. It then gets broken down by a protein called, or an enzyme called dicer. And then that dicer allows it to enter the nucleus. Once the mRNA is, once the double-stranded RNA is in the nucleus, it can then split apart into ligands, right? Into its single-stranded complements. Then proteins called the RISC complex can develop kind of like protecting the RNA around it. And then what happens is at the same time, you have transcription happening in the nucleus. Once transcription happens in the nucleus, this protected mRNA kind of like rogue agent, the secret agent of RNA, binds to the complementary partner on the cellular mRNA, which targets it. Once it binds, it makes that complement, it's targeted for destruction by the RISC complex, that protein complex. So it's a really complicated mechanism, that RISC complex. So what we're going to do is we're going to watch a quick video, and then we can look up a video on cytokine storm.